Hello, listener. Welcome back to Belharra, the birth story of a biotech podcast. I'm Jeff Yonker, the lucky guy who gets to host this podcast, and the even luckier guy who gets to be CEO of Belharra Therapeutics. I'm particularly pleased to be joined by my three guests today. Sherry Niessen, Belharra's head of platform and proteomics, Sean Buchanan, our head of biology, and Justin Ernst, our head of chemistry. Now, these three work as a unit. Think of them as three musketeers or three amigos or what have you. So it's particularly appropriate to be talking to them today as a trio. We'll learn a little bit about the individual journeys they took into science and then industry and then ultimately to a brand new startup. And we'll talk about how they work together and how their teams work together to advance our mission to develop game-changing medicines for patients in need. For any budding scientists out there or folks just wondering what it's like to be one of the very first to parachute into a research stage biotech startup, we've got a great episode in store for you as we climb into the gears of a high-performance research engine with Sherry, Sean, and Justin. So with that, thanks for joining me today, and let's go. It's like three legs of a stool, and the top of the platform of that stool is our drug discovery, and each of you represents a function that is one of those legs. You can't have any one leg be out of sync or significantly out of scope with the others, and so we're going to hear a little more about how we get it done here. But before we get to that place, want to get to know each of you a little bit better, kind of help our listeners kind of understand where you're coming from and kind of what you do. But why don't we start out with this question? So when was the first moment that you knew you wanted to be a scientist?
I was really into insects and the natural world, so I knew I wanted to be a biologist of some description at, I don't know what age, 6, 7, something like that.
All right, it's pretty young, yeah.
I would catch spiders, have them in my bedroom.
Yeah, I was a similar age. I had a chemistry set.
Oh.
Probably
That's almost
illegal
too predictable,
nowadays.
Justin.
yeah, but playing with chemicals out on the picnic table.
Nice.
Yeah.
Nice. Not blowing things up.
Well,
yeah.
I had a Bunsen burner, amazingly. Yeah, so
That's cool.
Back when they still had those
So
things.
complete chemistry kit.
Yeah.
You had your test tubes
Yeah,
and
by
Bunsen
mail.
burner and all the rest.
Yeah.
Excellent. So about same age then you were.
Yeah, around like 5 or 6. Yeah.
Yeah. And Sherry, what about you?
Actually, pretty similar to Justin in that my dad had made a room in the basement in the back of the house. And in it, I set up a chemistry set that I got from Sears and a biology set, I think, that I'd also gotten from Sears for Christmas.
You were a chemical biologist from the beginning.
She was.
Exactly. It had little butterflies. You could look under the microscope.
Yeah.
All sorts of chemicals you could mix together, do small experiments.
Nice.
My dad had his workshop in the basement as well, so you could mix it with, gasoline or other substances that you might find
Hypothetically.
in his
Gunpowder.
workshop could mix with my workshop.
Excellent.
Yeah.
That's fantastic. And did either of you or any of you sort of vary in that sort of aspirationity point? Anybody want to become a race car driver or something different along the way and then come back to science, or has it always been that you're true north?
Most certainly science. It may have fluctuated between marine biology, chemical
Right.
engineering,
Yeah.
then realizing I wanted to come back more closer to the biology type of sciences and then biology and so on.
Yeah.
But always science.
Always science. Yeah.
Yeah, my first love was physics.
Ah.
I was at Penn State, and I toured the physics department and then the chemistry department. And I said, I'm going to switch to chemistry.
Yeah. They're having more fun over there.
What was wrong with what was wrong with the physics department?
Just some strange individuals. Less strange people in chemistry.
Well, okay, all right. Well, we don't employ any physics at this point, sir. Physicists at this point, so I guess that's. So I'd love to hear. Each of you is somewhat unique on the team in that you bring substantial industry experience to this job. We have a lot of folks that are in their first jobs in industry. They come out of academic backgrounds. Most of your team members, actually, so you all know this. But I would love to hear a little bit about how you went from doing your PhDs and sort of made that move over to industry, and then what that path has looked like.
Yeah, actually, I started the industry in a really interesting way. So my first company was based off of some of my graduate work at Yale University. Mm-hmm. So we were focused on designing proteomimetics targeting protein-protein interactions. Mm-hmm. And the professor I was working with, Andrew Hamilton, formed a company with a couple of Scripps professors. Mm-hmm. And we decided to have the company out here in San Diego.
To your great fortune, because New Haven is almost as balmy of a climate. Yeah.
It's a growing biotech community there, but it made more sense from a business perspective to put it out here. And so I had the opportunity to kind of spin out with a company for my graduate work. So that was pretty exciting.
So that was no brainer then. You were just off and running in the industry and made that sense to.
What a great way to start the business.
Yeah.
So for many of the listeners of the podcast, I did my PhD with Ben Cravat. And so during my PhD, I became very passionate about developing and applying technologies to understand biology or to discover chemical probe starting points for proteins of interest. So as my PhD was wrapping up, we sort of talked about an opportunity to stay in academics, create a small center for chemoproteomics that would support academic researchers. It seemed like a great idea. I would provide unique chemical tools and opportunities, support grants, drive biology. Yeah. After 4 or 5 years, I wanted more of a translational impact, and a position opened up at Pfizer in La Jolla. I was, in fact, their first full-time hire, their first chemical biology hire. Okay. I joined a medicinal chemistry group, which is a really great position for a chemical biologist such as myself. Got to work with leading chemists to make tools, and then I would apply them for oncology. Yeah. And so that was, a great first position where I spent, the next 10 years doing that type of science.
So you've all seen a number of different organizations, some big, some small. You came to Belharra, now had almost 2 years, I think, in a couple cases, more than a year. So how would you compare and contrast? Like what is sort of consistent with the way that we are at Belharra with other environments you've been part of, and what's sort of distinct or different about the way that this company is and operates?
I think I would say I have been lucky in my 10 years at Pfizer and then my time at Belharra that we're all focused on patients. They're good science, the right science. Yeah. My teams at both small and then large biotech, all are really aligned on a similar mission. So that has been a lovely experience. Yeah. I think what is unique here is the speed at which we do, and the speed at which we're moving our science forward. Yeah. The speed at which we want to move our science forward is also a little bit different. Yeah. So I think the good thing is
demanding CEO, so that
demanding colleagues. Yeah. So the science equally
3 legs of a
good,
stool. Come on, they're all
equally exciting. The speed at which we're moving here is much quicker.
Well, firstly, the speed is exactly right compared to certainly big companies. But one actual commonality that many biotechs don't have, I don't think, is the experience level that we have at Belharra. Yeah. It's a pretty experienced team for the size of the company. Justin's 20 plus years in industry. Sherry's got phenomenal industry experience. I'm 20 some years in big and small companies. Yep. With Gary, who I think has done an early podcast,
track of the years,
we've stopped keeping track of the years. We've got a phenomenal experience level there, and in other parts of the company. So I think that's a commonality, but also a difference from other biotechs.
Yeah, I think one thing that's different is for a small biotech, we have the opportunity to build a pretty large platform. Yeah. And that's somewhat unusual from my experience. So it's just a great opportunity to build something big, different, that can really drive the drug discovery process. Yeah.
So a couple things that came out of a conversation that we had with Chris and John, 2 of the co-founders. John actually had an interesting analogy that maybe, just maybe, if you think ambitiously, what we're building here is akin to the iPhone. And the reason he made that analogy was said, look, that no single element of the iPhone was unique. It was really the combination of them all and the way they were combined. And you could arguably say the same about our platform, right? There's no element that has not existed somewhere else before, and it's really the way we combined them and kind of the way we're operating them that seem to be breaking ground so dramatically. Curious, what do you all think of that? Does that resonate, or would you take exception to it, or how do you think about our platform, right, in air quotes?
Yeah, I view it as it's an immensely powerful platform, and as Sean pointed out, it's amazing to see how it's playing out with a group of experienced industrial veterans and really leveraging that with the intent to make drugs. Yeah, and Sherry, you've been part of building a couple platforms now, so I mean, it's. Do you think that rings right? That maybe the way we're doing things is sort of different enough that it may be.
I mean, even if you think of some of the different technologies that we are applying, I don't think 10 years ago they existed with the same speed and capacity at which they are available today.
Yeah.
And we can sort of leverage those opportunities to build this platform. So we can go fast, so we can profile a really great chemical probe library. So I think it is probably a really neat analogy.
Sean, anything more you'd add on that?
I don't think so. I like the analogy though.
Yeah? Well, I love the ambition.
Don't tell
behind
John
it.
I said that. But
Yeah. All right, so we're going to do a test here to see how well you highfalutin scientists can bring science down to the most basic level. What I'd love for you the three to do, and it's a bit of a teamwork challenge too, is I'm going to give you 60 seconds. I want the three of you to describe how we're going to take a program from initial idea to a drug candidate that's ready to go into sort of preparation for human testing. And so just step by step, and you can sort of ping pong as you want. How do we do it, and how's this platform going to run from A to B on that? And I'm going to start with Sherry because that's where it starts, right?
I was going to say we should start with Justin because he makes the probes, and then he punts it to me.
Yeah, so for the listeners, I think what's unique about Belharra is we utilize a proprietary probe library. So these are drug-like molecules that have unique features on them that enable the platform. So we've gone ahead and built this platform from scratch, and Sherry's group then takes that library and screens it with her proteomic platform. So maybe you could describe that, Sherry?
Exactly. So we utilize the library created by Justin and his team, and we employ automation, quantitative mass spectrometry, and bioinformatics to profile those chemical probes to identify starting points and novel druggable pockets, which we then share with Sean and his team.
Yeah. And so where we are now, we have a phenomenal number of these proteins, pockets with drug-like molecules bound, and the biology group helps filter through these proteins to understand which of these might make a genuine impact on a disease. And our initial focus is immunology and oncology, and we've got some pretty exciting target proteins from this proteomic effort that we are busily rushing forward to see if we can find true medicines to treat aggressive forms of cancer and autoimmune disease. And the biology team helps develop the assays that are needed to translate these starting point molecules, support chemistry to turn them into drug-like starting points for clinical trials.
So at a high level, that sounds a lot like other screening platforms. And I think in purpose it is, right? I mean, we're all sort of chasing the same vision of can you find a place that a drug can actually bind and get modulation of a protein for therapeutic benefit. You all made an important choice to join this place. You left other opportunities that were particularly good and probably turned down others that were even, as good. So what got you here? What's the conviction behind that explanation you just gave me? What was it? It was like, oh yeah, I got to be part of this, because I think great stuff's going to happen.
It's a unique platform. Yeah, it's a truly unique platform. All of us have been around the industry for a while. These platforms come every once in a while, but this one is truly special. It's got incredible potential. So coming to Belharra was an opportunity to deliver on that potential, but also, so that's one, and also to work with a phenomenal team. It's both the experience, but also just the group of people, the CEO and the CSO who started this all as the two founding employees, I think, of the company. So those two things, kind of rare to find despite the proliferation of companies out there.
Yeah, I had sort of a unique opportunity because my prior position was head of chemistry for a company called Inception Therapeutics, which is an incubator working with Versant Ventures to build biotechs. And this was one of the companies that was pitched while I was there. So I had the opportunity to review the science and then helped Chris, John, and Ben sort of build the company along with the other Inception employees to the point where we spun it out in October of 2021. But the concept was originally pitched in early January of 2021, and yeah, it was just really exciting to see it grow from scratch. And just I love the fact that it was a chemistry-based platform. Love the fact to work with Ben and Chris, and yeah, just really jumped at the opportunity to move over with the company.
Yeah. So I think we'd probably say it was the founding ideas and then the leadership and support we've gotten to build the platform,
We probably
which is unique.
It is, and we probably haven't said much, at least in this podcast, about quite what it is that's unique about the platform. Like, like for me, it's the opportunity to do something different on notoriously difficult problems in drug discovery.
Mm-hmm.
Maybe Sherry can say something about why this platform has a
It's true.
chance to make
do
breakthroughs that others have not yet.
I think so. For many of us that have been in traditional drug discovery, you really start with proteins that are assayable. This platform allows you to tackle tough to drug targets because you can screen them in live cells. So when they're in their most natural, endogenous constraints or confines. So we can screen our chemical probe library to identify these starting points in that particular environment.
So in that sense, the cell becomes your assay system.
Yes, when they're more assayable.
Right. And why is doing this all in the cell system so important?
Yeah, I think it's partly what Sherry just said. Many of these proteins have been tried in a so-called cell-free environment, and not much to show for it for some of the most difficult targets in the industry. But it's
so pause on that for just a second, because is it a problem that this company is going after a whole bunch of targets that everybody else knows about?
We're doing it in a very different way. We're doing it in a very different way, and so where others have struggled, for sure, and I've been part of those missions, we're trying something truly different. So yes, we don't yet know how often we will succeed, but although the early data are super promising, but it's a very different way of doing it, and we're doing it in the native system. We're doing it in human cells, typically, that represent the disease we're trying to treat. That's not easy to do with any other methodology, or can't be done with almost any other methodology. And we've already got starting points that we know you would not have found the conventional way.
Yeah. We talk about having a vision that we have a platform that sort of can see any binding pocket, any protein, any conformational state, any cell type, and we say that a lot. What does that mean? If we've so far all the data support that statement, we hope we'll continue to build a data set that sort of further proves it. But even if we only come close to that, it's still a pretty remarkable statement. So why is that significant? What does that put us? What position does that put us in as a platform relative to so many out there?
Yeah, a lot of the other chemoproteomic platforms focus on using what's called electrophilic chemistry, which requires reaction with certain specific amino acids on proteins. So that limits the types of proteins that one could target with those platforms. Our chemistry is agnostic to which proteins the molecules interact with, so which opens up the possibility of targeting any protein in the proteome.
And it also, I think that one of the things I think we've talked about, Sean, is the notion that some of these pockets may not even be visible unless you do see a protein in this sort of functional state and its interaction with other proteins, or sort of signaling the way that it needs to. Is that part of what you think we're already seeing with some of the data that we're generating?
Yeah, I think we have good evidence that that's exactly what we're seeing in some cases, and it builds on what Justin pointed out, that this method is truly unbiased. Prior approaches are sort of biased to what's known about the pockets that are known to exist, but of course everyone realizes there's at least as many pockets that we don't know about, and we have a method to discover those in a truly unbiased way. We're not limited by the amino acids in the pocket context, for example, as Justin pointed out.
So platform is working. It's generating a lot of interesting target ligand interactions or hits that we can start with across a bunch of targets that we already have a bunch of biological evidence are likely meaningful for human disease. And how do we pick now? How do we start prioritizing? That's where I know the three of you spend a lot of time in discussion.
It certainly starts with the importance of the protein target in disease. So our North Star is trying to find truly transformative medicines. And we know, in immunology oncology, we know of many proteins that if only we could find a decent drug, it's fully expected there would be transformative benefit to critically desperate patients. So that's the North Star, and we can rank targets based on the evidence that that's the case. So that's the starting point. And we're lucky enough with the platform so far that we're finding ligands or probe hits for many of these super important targets that had just not been druggable previously.
So talk a little bit about where we are headed. Sean, you just sort of described it as your North Star, our North Star, which is delivering game-changing medicines or high-impact medicines. We're early stage, right, as research goes, and many miles to go still into the clinic and beyond. How do you keep that focus, that long-term vision of what we're really here about, in sort of the way you think about your function or the way you lead your teams?
How do we keep them focused? Deadlines.
Well, drug discovery is a marathon. Every step of the way, you have to be aware of the present, but always focused on, the overall game.
Yeah.
And we have a lot of experienced individuals here that have put many molecules into the clinic, delivered drugs. So I think it's that experience with just the long-term vision for the company and the platform.
Hal Barron, who was the chief medical officer at Genentech back in the day, used to say that we were the world's most optimistic people, right? Mm-hmm. To show up every day at work knowing there was a 99% failure rate, and that believing your program wasn't going to be that one, or was going to be the one, I guess. So it's motivating and focused, but it's also reminding people that there is this sort of higher purpose that we're about, that it's not just building a platform, it's not just running screens, it's not just identifying chemical matter. There's actually a much bigger arc that we're playing for, which is someday a physician dosing a patient and their lives being changed by it.
When I was at Vertex Pharmaceuticals, one great aspect of working there was they often bring in CF patients, the majority of which are younger individuals. Yeah. And they would talk about the power of the Vertex medicines for changing their life. A kid that couldn't play soccer, that can now play soccer. We brought in a teenager that used to sing opera, and she had to give it up, and then she started taking the Vertex drugs and then could sing again. So that's pretty motivating.
That thinking instills a sense of urgency. Sherry was saying at the outset how a difference between this company and many others, certainly the larger companies, is the speed with which we can do things. And there's no reason why we can't pretty quickly get to that point. Pretty quickly have medicines, seeing patients and see what they can do. And that's what's driving, I think, all of our teams, right?
Yeah. Let's talk about sort of the how you build the environment and the culture of a place, because that's something that I know each of you spends a lot of time on. How would you characterize Belharra today, and sort of what do you think about the way we are, the way we operate, is impacting the science we're doing?
I think we brought in some of the best people to do their particular jobs, some of the best people in automation, some of the best chemical biologists. And when you unleash those people, you get great results.
We have a lot of energy. It's a work hard, play hard environment for sure. It's a special culture.
Yeah, I think it's also embracing just being fearless. So I think we're not afraid to take on tough challenges, and just really having that mindset.
So maybe go back to this idea of sort of unlocking or unleashing people, and because I do think that's one of the things that we're trying hard to do, and I think using a lot of intentionality and seeing what will work to do that, because you've got very bright, very experienced, very motivated people, and so how do you even tune that up and sort of get everybody to really pour it on? And you're clearly doing that. And so I'm curious sort of what's your take on the recipe here that's sort of enabling that?
Well, at least for my team, when I recruited in each of the different members, which really do have diverse expertise, whether it's quantitative proteomics or informatics or automation, I tried to recruit in people who are better at all of these types of experiences than I am. Right, so that was key. So my job is really to kind of focus their energy and focus their talents on our goal. And I think in part they achieve it just with their own desires, whether it's impacting patients or whether it's building the platform. I truthfully think they all enjoy each other's company, and I do think that helps in this recipe.
Yeah, we have a very passionate group of people. Everyone is working on a single team. Yeah, it's a Belharra team.
Like an aligned
that's
mission.
Obvious.
Yeah.
We're all
when
aligned.
you walk in the door, and you know, sort of sounds obvious, but it's a really important aspect of why you're seeing what you're seeing, Jeff. And
Well, we've all been in environments where that wasn't always the case, right?
Yeah.
And when you have misalignment, you know, people are pulling in the oars, but in different directions.
Yeah.
I mean, it's chaotic, right? And unproductive.
Yeah.
So,
Yeah, I think the teams believe in the vision of the company.
yeah.
yeah.
They believe in each other and in the vision of the company, and they want to work with each other.
Yeah.
Yeah, they're all nimble too.
Yeah.
So if there's an obstacle or we learn a piece of data, we're all very quick to be able to change, adapt, and then continue to move forward.
I want to talk a little bit about where you hope we go, and maybe take it far enough down the road that it sort of maybe uncouples a little bit from the exact things that we're doing right now. But if you think sort of 8 to 10 years from now, and what something like this might be able to do.
I hope to identify a chemical probe for every protein in the proteome. To have
Wow.
a chemical probe starting point for every possible pocket and protein.
That's awesome.
Sean could have his selection of opportunities to follow up on.
Sky's the limit.
Within 8 to 10 years, hopefully we have some really outstanding molecules in the clinic that are impacting disease.
Yeah.
I think that's an achievable goal within that time period.
Yeah.
That for me is also the driving motivation for, I think, the entire team shares that vision and the belief that that's what we can do.
Yeah. So last question for each of you, and then I think we'll wrap here. Is so when you're not at Belharra, and what's your happy place? What are you doing? What are you enjoying?
I love spending time with my family. I have a great wife and 2 young daughters. Love to play golf. Just like drug discovery, golf is extremely challenging. But when you hit that one good shot, it's addictive and you keep coming back to it. So
The 1 out of 100.
so I'm
Right?
chasing the dream.
Perfect.
Yeah. Sean, how about you?
Two things I think I do in my spare time, both with my family, if I can convince them. One is spending a lot of time outdoors. Love the wilderness, as remote as possible.
Nice.
That's me, and luckily the rest of my family kind of tags along. They're less inclined to tag along to the rugby game, so that's my other big passion, and so happens that the Rugby World Cup's just started, so if you guys don't see me on occasion during the workday, it could probably mean there's an important rugby game going on. But
Excellent. We'll get it on the TV in the break room.
yeah, we did actually the other day.
Good.
Yeah.
Sherry, what about you?
Outdoors, hiking, exploring new trails in San Diego, traveling. We did a recent trip to Italy, and so the family and I have been very focused on trying to make a reasonable pasta sauce. Inspired by a cooking class that we did.
Which particular one? Which
Just tomatoes.
Okay, yeah. All right.
Olive oil, garlic, a whole bunch of different types of tomatoes, sort of boiled down into a really nice sauce.
Yeah.
We've been practicing, and we're getting better.
Are you thinking fresh or canned tomatoes? Which way are you leaning?
Oh, fresh.
Fresh
For sure. Yeah. So we're going to perfect our tomato sauce one day.
Sounds excellent. I want to thank each of you for a lot of things, particularly being here and just giving all you do and kind of putting your all into this company and what we're doing. But also for building the teams you have and leading them the way you do. And for spending time today, taking time out just talking about this. So thank you very much.